imagine for a second that your body is fighting this massive war against a tumor.
Right.
If you look at standard textbook immunology, we usually picture the immune system acting like a distant, centralized command center, right?
Exactly. It dispatches troops through the bloodstream.
Right, sending them from the lymph nodes to this hostile, faraway battlefield. But looking at the latest research, that systemic view is, well, it's starting to look a little outdated.
It really is.
Because what if, instead of just sending soldiers from hundreds of miles away, your immune system had the actual engineering capacity to build a fully functioning, heavily fortified military base right inside enemy territory?
I mean, it sounds totally counterintuitive, right? Especially since we are so used to thinking of the tumor microenvironment as this, incredibly immunosuppressive wasteland.
Yeah, wasteland.
But building these localized bases is exactly what happens in patients who successfully fight off cancer. They construct these highly organized immune hubs directly inside the tumor stroma.
And exploring how they do that is our mission for today's deep dive. We are unpacking this phenomenal 2026 research article published in Science by Matthias and colleagues.
Such a great paper.
It's titled Dendritic Cells Control Tertiary Lymphoid Structure Development and Maintenance in Cancer. So we're going to dissect this paper right down to the studs to understand the mechanics of how the immune system constructs these secret bases.
And the specific architect cell pulling the strings.
Yes, exactly. And how understanding this architecture is already shifting the paradigm of cancer treatment.
Because the clinical stakes here, I mean, they really can't be overstated. To understand why immunologists are so laser focused on this, we really have to look at the human data driving it.
Right.
The Matthias paper anchors its findings in major clinical trials, specifically the Popular trial, which looked at patients with non-small cell lung cancer.
NSCLC.
Right. And they compared standard chemotherapy to an immune checkpoint blockade, specifically a atezolizumab.
Right. And just as a quick refresher for everyone listening, immune checkpoint blockade, or ICB, essentially takes the brakes off the immune system. Drugs like atezolizumab block the PD-L1 pathway, which tumors use to sort of hide from T cells.
Exactly.
But looking at the Popular data, I was just struck by the variance in patient response. I mean, not everyone benefits from taking the brakes off.
No, they don't.
Yeah.
Because taking the brakes off, well, it only works if you have a functioning engine and wheels on the ground.
Right.
When the researchers analyzed the patient's tumor biopsies, the single greatest predictor of overall survival wasn't just the sheer number of T cells present. It was spatial organization.
Wow.
Yeah. The patients who survived the longest had high densities of these localized immune bases, which we call tertiary lymphoid structures, or TLSs.
Okay.
And this wasn't an isolated finding either. They saw the exact same pattern in a hepatocellular carcinoma trial using a totally different ICB drug, semiplimab.
Okay, so if these tertiary lymphoid structures, these TLS bases, are the ultimate predictor of surviving lung and liver cancers, it begs the obvious question, right? Why doesn't every patient develop them? Like, who is initiating the construction, and why does the process just stall out in the patients who don't respond to therapy?
Well, to get to the bottom of that, we really need to look at the cellular anatomy of a TLS. So you have normal lymph nodes, right?
Right.
You're born with those. They develop during embryogenesis, and they just sit in fixed anatomical locations. A tertiary lymphoid structure is entirely different.
It's makeshift.
Exactly. It's a temporary makeshift organ that forms in non-lymphoid tissue strictly in response to chronic unresolved inflammation like an autoimmune disease, a severe infection, or in this case, cancer.
So it essentially functions as a forward operating base,
Right.
like a FOB. But unlike a disorganized inflammatory response where cells just flood the area and cause total chaos, a mature TLS is incredibly structured.
Oh, the level of organization is stunning. The researchers utilized some incredibly advanced spatial transcriptomics here, technologies like Visium-SD, MERFISH, and CiteFIF.
Right, and for those of you not working in a pathology, these tools basically allow scientists to map messenger RNA expression at single-cell resolution while keeping the 3D architecture of the tissue completely intact.
Exactly. And by doing this across human, lung, liver, colorectal, and renal cancers, they prove that a mature TLS isn't just, a clump of cells. It has a distinct B-cell follicle and a highly active T-cell zone.
Right, and the cellular roster inside these zones is just highly specialized. Like, looking through the MERFISH data in the paper, you see T follicular helper cells, TFH cells, which act as the localized commanding officers for the B-cell responses.
Yes. And right alongside them, you have the TPEC cells.
Okay, the TPEC cells.
Yeah, which stands for TCF1 positive, PD1 positive progenitor exhausted CD8 plus T cells.
Okay, that is a massive mouthful of immunology jargon.
It really is.
But functionally, TCF1 indicates a stem-like state, right?
Precisely. So while they express PD1, which indicates they've seen the tumor antigen, that TCF1 marker means they aren't fully exhausted or terminally differentiated.
I see.
They retain their proliferative capacity. So they're essentially this elite reserve of killer T cells resting inside the base, just waiting to be deployed to the tumor bed.
And you also have the germinal center B cells, right? The spatial mapping showed these cells are AID positive, meaning they are actively undergoing somatic hypermutation.
Yes, exactly.
So they aren't just generic immune cells. They are literally a localized weapons factory manufacturing highly targeted tumor-specific antibodies right on the front line.
Right on the front line.
But none of this machinery works without the cell at the very center of the paper's thesis.
Right. The absolute linchpin of this entire operation is the CDC1, the type 1 conventional dendritic cell. If the TLS is a forward operating base, the CDC1 is both the chief architect and the lead intelligence officer.
The spatial transcriptomics visually prove this, too. Like, the data shows these mature CDC1s physically clustering right at the borders of the T and B cell zones. They are positioned as the central node, physically interacting with the other lymphocytes.
And that physical proximity, it's not an accident. They're coordinating the entire immune response.
Okay, so synthesizing this first piece of the puzzle for you guys listening, we are looking at makeshift, temporary immune organs built directly inside tumors. Their presence strongly correlates with cancer survival, and they function as highly organized bases packed with specialized T and B cells, all orchestrated by this central architect, the mature dendritic cell.
Exactly.
I mean, I love to picture this bustling underground city inside the tumor. The CDC1 is the architect standing in the center, handing out blueprints to the T cells and B cells. It really forces you to reconsider the engineering capabilities of your own immune system.
It really does.
Have you ever considered that your immune system has the engineering capability to build temporary organs on demand? It's wild. But, knowing what a TLS looks like is only half the battle. The core question Matthias and the team wanted to answer was how this base gets constructed from scratch in a seemingly hostile tumor microenvironment.
Which is an incredibly difficult thing to study in human patients.
Right, because of timing.
Yeah. By the time a patient undergoes surgery and a tumor is biopsied, the TLS is either already fully formed or it's entirely absent. You only get this static snapshot. So to catch the immune system in the act of building the base, the researchers had to utilize a highly specific in vivo laboratory model.
Right. They used a genetically engineered mouse model called KP-HELO2.
Yes.
And from my understanding of the paper, this isn't just any tumor model. It's a lung cancer model, specifically engineered to express distinct model neoantigens, and it predictably forms mature TLSs.
Exactly, which is crucial. It gives the researchers a standardized timeline to track the exact immune cells responding to the tumor day by day.
Okay, so it creates the perfect temporal window.
Right. And by watching this model closely, they mapped out a very rigid 3-step biological sequence that occurs during the first 8 days of tumor growth.
Okay. Let's break down those steps.
So, step 1 is maturation. Initially, the CDC1 architects are stationed in the tumor tissue in an immature state. They scavenge the area, phagocytosing or eating dead tumor cells, thereby taking in the enemy's antigens.
But just eating the antigen doesn't activate them, right? The paper highlights a critical dependency on interferon gamma.
Yes.
The CDC1 requires signaling through its interferon gamma receptor to actually wake up and switch into a mature active state.
Without that interferon gamma signal, the CDC1 remains functionally inert. It just sits there. But once it receives that signal and matures, it triggers step 2. Migration. The CDC1 radically changes its surface proteins, heavily upregulating a chemokine receptor called CCR7.
And CCR7 acts essentially like a molecular GPS tracker, right?
Yeah, that's a great way to put it.
It detects a chemical gradient that guides the CDC1 out of the tumor bed into the afferent lymphatic vessels, and it commutes all the way to the systemic headquarters, the tumor-draining lymph node.
And the reason for this commute brings us to step 3, T-cell priming and recruitment. Once the CDC1 arrives at the lymph node, it performs this highly specialized immunological feat called cross-presentation.
Okay, let's pause and dig into cross-presentation because this is where the biochemistry gets really elegant. Normally, a cell presents proteins it made inside itself on MHC class I molecules to activate CD8 killer T cells, right?
Right.
And it presents things it ate from the outside on MHC class II molecules to activate CD4 helper cells.
Exactly. It's a fundamental rule of immune compartmentalization. But cross-presentation breaks that rule completely.
Really?
Yeah, the CDC1 takes the tumor antigens it ate from the outside and somehow routes them into its own internal MHC class I pathway.
Wow.
And the researchers found that this specific routing heavily relies on a gene called WDFY4. Right. By using WDFY4, the CDC1 could display the tumor antigen to naive CD8 plus T cells in the lymph node, essentially handing them the exact molecular mugshot of the tumor.
That is so cool.
It activates them and directs them to swarm back to the tissue to start building the TLS.
And the researchers didn't just observe this happening. They proved it functionally by selectively breaking the system, which I love. They used genetic ablation models, specifically XCR1 DTA mice.
The genetic tools here are fascinating. XCR1 is a chemokine receptor uniquely expressed by these specific dendritic cells.
Okay.
And DTA stands for diphtheria toxin A chain. By genetically linking the DTA gene to the XCR1 promoter, the researchers engineered mice where every single CDC1 naturally self-destructs during development.
So the mice literally just have no CDC1s?
Not at all. And when they introduced the lung cancer model into these mice, absolute silence. The TLS never formed and the tumor grew completely unchecked.
Wow. And they also proved the commute itself was mandatory during this early phase using a drug called FTY-720, right? As I understand it, FTY-720 is a sphingosine 1-phosphate receptor modulator. Functionally, it locks down the lymph nodes, trapping the lymphocytes inside so they can't travel back to the tumor.
Exactly. It traps them.
So if they administered this drug during those first 8 days, the TLS construction failed entirely. The architect could make it to headquarters, but it couldn't bring the labor force back to the construction site.
It unequivocally proves that the initial seeding of the tertiary lymphoid structure is a systemic event. It absolutely requires the lymph node.
Okay, let me do a quick mini recap here. So in the early days, early TLS formation absolutely requires the CDC1 architects to mature via interferon gamma, upregulate CCR7, commute to the lymph node, utilize WDFY4 to cross-present the tumor antigen, and recruit T cells back to the tumor.
Perfect summary?
It's a massive logistical undertaking relying on systemic circulation. But I have to push back a little here. Looking at the scale of a progressing tumor, a mandatory continuous commute back and forth to the lymph node seems biologically incredibly inefficient.
Oh, it is.
As the tumor mass expands and the metabolic demand increases, relying on a microscopic commute to recruit every single new worker, it just doesn't seem scalable. Can you picture this microscopic cellular commute happening in your own lymphatic system constantly?
You've hit on the exact anomaly that drove the second half of this paper. The commute is biologically expensive. And what Matthias and colleagues discovered is a major plot twist.
Oh, I love a plot twist.
Around day 8 of tumor development, the systemic commute completely drops off.
Wait, it just stops?
The rules of engagement change entirely. It doesn't just shut down, it localizes. We enter what the researchers call the late phase.
Okay.
Through flow cytometry and spatial tracking, they noted a steep decline in the number of mature CDC1s arriving at the tumor-draining lymph nodes. The architects basically stopped commuting. Instead, they began pooling locally, taking up permanent residence inside the tumor stroma.
So they transitioned from commuting to headquarters to literally working from home, managing the war locally.
Exactly, working from home.
But they aren't just scattered randomly throughout the tumor, right? The spatial mapping showed them anchoring at very specific coordinates called stromal hubs.
Yes. These stromal hubs are microenvironments rich in structural cells, specifically cancer-associated fibroblasts, or CCAFs, and perivascular cells that wrap around the blood vessels.
Now, I have to challenge this slightly. Why are cancer-associated fibroblast cells that the tumor typically uses to build its own physical scaffolding and, shield itself, why are they creating a welcoming hub for immune architects?
It's an incredible example of the immune system hijacking the tumor's own infrastructure.
Really?
Yeah. These specific CCAFs and perivascular cells pump out a chemokine called CCL19. Do you remember how the CDC1s upregulated the CCR7 receptor to navigate to the lymph node?
The molecular GPS tracker.
Yeah, actually. The lymph node naturally produces CCL19 to attract them.
Oh, I see.
Right. But as the tumor develops, these localized stromal hubs start producing massive amounts of CCL19 as well. They essentially create a decoy lighthouse.
A decoy lighthouse.
Yeah, the CDC1s, still expressing CCR7, they follow the gradient, navigate directly to these paravascular hubs inside the tumor, and just drop anchor.
So they use the tumor's own stroma as the physical foundation to build the forward operating base. That is so smart. But just parking themselves at a stromal hub isn't enough to maintain a complex immune structure. Like, what are they actively doing to keep the T cells and B cells engaged?
This is where the paper uncovers a profoundly complex molecular mechanism. To prevent the TLS from dissipating, the CDC1 engages in continuous, localized antigen presentation. But it isn't a simple handoff. The researchers found it requires a simultaneous three-way biochemical triad.
Okay, let's break that triad down.
The CDC1 must present the tumor antigen on MHC class I molecules to the CD8 killer T cells, while concomitantly presenting antigen on MHC class II molecules to the CD4 helper T cells.
Concomitantly, meaning it acts as a simultaneous molecular docking station for both the killer and the helper T cell lineages at the exact same time.
Yes, at the exact same time. But even that dual docking isn't enough. No. While locked into this presentation, the CDC1 also requires a distinct validation signal back from the CD4 helper T cell, specifically via the CD40 receptor. It is a continuous reciprocal feedback loop. Antigen goes out via MHC-IN2, and survival signals come back via CD40.
Wow. So to keep the base from collapsing, the dendritic cell literally has to stand in the middle, holding hands with a CD4 T cell and a CD8 T cell at the exact same time, constantly validating their mission.
That's a perfect way to visualize it.
If we think of this in terms of structural engineering, it's like a keystone in a cellular arch. The dendritic cell is the keystone, physically bridging the CD4 and CD8 compartments. If it drops either connection, the architectural integrity fails.
Absolutely fails.
And again, the researchers proved this through incredibly precise genetic engineering, this time using the XCR1 DTR model.
The DTR model is crucial here. Unlike the DTA model we discussed earlier, which deletes the cells constitutively from birth, DTR stands for diphtheria toxin receptor. This system acts as a genetic remote control. The mice develop perfectly normally, the CDC1s are present, they do their early commute, and they successfully build the TLS during the first 8 days.
But then the researchers flip the switch. On day 8, they inject a highly localized, minute dose of diphtheria toxin. And because only the CDC1s have been engineered to express the receptor for it, the toxin exclusively assassinates the architects, leaving the rest of the immune system completely untouched.
Exactly.
So what happens to the base?
Total catastrophic collapse. Within days of removing the local CDC1s, the TLS completely dispersed. The T-cell zones fragmented. The germinal centers, the weapons factories making antibodies, they vanished entirely. The production of tumor-specific IgG antibodies plummeted. The base simply ceased to exist.
That is insane. And they took this a step further, right? They didn't just delete the whole cell. They engineered mice where the CDC1s were functionally intact, but selectively missing only the MHC class I molecule, or only the MHC class II molecule.
And the result was exactly the same. If the CDC1 couldn't maintain that simultaneous molecular docking station, if it couldn't engage both the CD4 and CD8 T cells concurrently, the local TLS dissolved.
Okay, so synthesizing this crazy shift for everyone, we move from a systemic lymph node-dependent seeding phase into a highly localized maintenance phase. The CDC1s hijack the tumor's CCL19-producing fibroblast to drop anchor and then act as a structural keystone, locking into a simultaneous MHC-I, MHC2, and CD40 feedback loop to keep the entire immune base from crumbling.
It's beautiful, really.
The sheer biological precision required for the shift from remote control by the lymph node to local control inside the tumor is staggering. What stands out to you listening to this about this incredible shift? It's just wild. But as fascinating as the biology is, the ultimate goal of understanding this architecture is to weaponize it for patient therapy.
Exactly. We are transitioning from discovery to clinical application now. If the maintenance and expansion of these vital tertiary lymphoid structures rely completely on the local abundance and active signaling of CDC1 architects, well, the most logical therapeutic intervention is to engineer a way to flood the tumor microenvironment with more of them.
Which is exactly what they tested in the in vivo models. The researchers administered a drug called FLT3-LFC. FLT3 ligand is a well-known growth factor that specifically drives the hematopoiesis, or the production and expansion of dendritic cells.
When they administered FLT3L on day 8, precisely when the TLS shifts to relying on local architects, it dramatically amplified the immune response. It successfully doubled the density of TLSs inside the tumors.
So they essentially airdropped in a massive surplus of structural engineers?
Yes. And they combined this strategy with a CD40 agonist. Since we know the CDC1 requires that CD40 validation signal from the helper T cell to stay active, researchers bypassed the T cell entirely and artificially provided that signal using an agonistic antibody.
Oh, clever.
Very. This drastically expanded the TLS networks, but, and this is a critical caveat, it only worked if the CDC1s were physically present and capable of antigen presentation. You can't just spam the tumor with activating signals. The physical architect must be at the stromal hub to receive and translate that signal into structural maintenance.
But curing cancer in tightly controlled, genetically identical mice is one thing. Translating a multi-layered immunological mechanism into human patients is historically where, well, a lot of promising therapies hit a wall. Does this architecture hold up in the clinic?
It absolutely does, and the Medias paper provides a really compelling real-world translation here. They detail a patient with metastatic breast cancer who was treated with a highly experimental in situ vaccination protocol.
Okay, in situ.
Right, so this isn't a traditional prophylactic vaccine injected into your arm. This was a complex, three-part therapeutic cocktail injected directly into the tumor bed.
Let's break down the mechanics of that in situ vaccine because it maps perfectly onto everything we've just discussed.
Right.
First, they administered FLT3L directly into the tumor to recruit a massive wave of dendritic cells to the site.
Second, they subjected the tumor to low-dose, localized radiation.
Yeah.
Radiation induces immunogenic cell death.
Right.
It acts like a localized explosive, rupturing the tumor cells and releasing a massive flood of tumor neoantigens, the raw building materials, for the newly recruited dendritic cells to scavenge and cross-present.
Finally, they injected a compound called poly-ICLC. For our listeners focused on molecular pathways, poly-ICLC is a synthetic, double-stranded RNA molecule.
Yes.
It is specifically designed to bind to toll-like receptor 3, or TLR3, on the surface of dendritic cells. Functionally, it tricks the dendritic cells into believing they are in the middle of a massive viral infection. It forces them to rapidly mature, upregulate their costimulatory molecules, and just get to work.
You provide the architects, you provide the building materials via radiation, and you provide the activation signal via TLR3. And the clinical results were striking. Biopsies taken from the human patient post-treatment showed a definitive, histologically confirmed increase in localized TLS formation.
That is just phenomenal. We are moving so far beyond the initial era of immune checkpoint blockade. We are no longer just broadly taking the brakes off an exhausted immune system and hoping for the best. We are learning how to drop supply crates, molecular blueprints, and artificial activation signals directly to our cellular architects so they can build more bases.
Synthesizing the entirety of this deep dive, the mechanistic through-line is incredibly clear. The presence of mature tertiary lymphoid structures fundamentally alters a cancer patient's prognosis.
Without a doubt.
And thanks to this work in science, we now definitively know that the mature CDC1 is the master orchestrator of this process. It dictates the systemic seeding phase by commuting to the lymph node via WDFY4 CRAS presentation. And it solely dictates the local maintenance phase by dropping anchor at CCL19 hubs and locking into a continuous multi-receptor triad with T cells.
It is an absolute masterclass in cellular logistics and spatial biology. And exploring the sheer complexity of this mechanism leaves me with a final, somewhat provocative thought to chew on for you guys.
Let's hear it.
In oncology, we constantly frame cancer as a disease of profound immune suppression. We think of the immune system as being beaten, chemically exhausted, or actively turned off by the tumor microenvironment.
Right, that's the standard view.
But when you look at the insane multi-layered structural engineering required to build and maintain a tertiary lymphoid structure, maybe the immune system isn't always fundamentally suppressed. Maybe it is just facing a massive localized supply chain bottleneck.
That is a very compelling paradigm shift. It reframes the immune system not as defeated, but as logistically starved.
Right. The T cells are clearly willing to fight. The CDC1 architects clearly know how to interpret the blueprints and build the base. But as the tumor scales, they simply run out of localized materials, or the biochemical triad gets physically blocked by the expanding stroma.
Yeah.
If future immunotherapies can act as targeted logistical support delivering FLT3L to recruit architects, radiation to supply antigens, and TLR agonists to simulate urgency, could we eventually train our bodies to turn any solid tumor, no matter how aggressive, into a self-destructing immune fortress? I want you to think about that as you review the latest clinical trial data in your own fields.
It's a great question to leave on.
How many different targeted therapies could be spatially combined based on this one dendritic cell mechanic? Keep questioning the underlying architecture of your own biology because the precise blueprints to fighting disease are quite literally written in the spatial organization of your own cells.